Okay, let's dive in. Forget everything you think you know about light reflecting off surfaces, coming from external sources. Today, we're plunging into the incredible, often hidden world of living light, bioluminescence.
Living light. It sounds almost magical, doesn't it?
It really does. We're talking about organisms that don't just use light, they conjure it, crafting it molecule by molecule from within their own bodies.
Hmm.
And here's a fact that genuinely stopped me in my tracks when digging into these sources. A staggering majority, we're talking over 75% of the creatures in the deep, dark ocean possess this ability.
That number is just astounding when you first hear it.
It is. It's not the exception down there. It's like the norm. It's like the deep sea isn't just dark. It's a universe dotted with countless tiny moving lights.
Exactly. For us, light is usually something you switch on or, something the sun provides. Imagine a world where you are the light switch and your body is the bulb. That's the reality for so many organisms.
Right. And that striking prevalence in the deep ocean is just the beginning of the surprises. Our mission today is to take this stack of sources you've shared. We've got everything from foundational scientific journal articles explaining the chemistry,
Real nuts and bolts.
cutting-edge reports from marine research institutions revealing where and why organisms glow, to reviews detailing its explosive growth in medical research, and even insights gleaned from direct observations, some captured on social media.
A really diverse set of perspective.
Definitely. So we're going to unpack what bioluminescence actually is, where we find it, explore the diverse and often ingenious reasons organisms employ it, delve into the fascinating biochemistry and genetics behind it, and then pivot to understand how this incredible natural wonder is being harnessed for significant advancements in medical research right now and, well, into the future.
It's a journey, really, from the deepest, least explored parts of our planet to some of the most advanced biomedical laboratories, all linked by this single remarkable trait. It truly showcases how fundamental curiosity about the natural world can yield unexpected, powerful tools for human health.
Okay, let's start at the beginning then. At its most basic level, what is bioluminescence? What's the science behind living light?
At its heart, bioluminescence is a chemical reaction. The word itself gives you a clue. It's a combination of the Greek word bios, meaning life, and lumen, meaning light.
Bios and lumen, life light.
Exactly. So it's literally light produced by living organisms. But unlike combustion, like burning wood or a candle, which generates a lot of heat along with light, bioluminescence is incredibly efficient. It produces minimal thermal energy, which is why it's often referred to as cold light.
Cold light. So none of that energy is wasted as heat. That makes sense from an evolutionary perspective. You only want to spend energy on the light itself if that's your goal.
Precisely. You want function, not just heat.
How does that chemical reaction work within the organism? What are the key players?
The key player is a molecule called luciferin. Think of this as the fuel, the substance that actually gets excited and emits light.
Luciferin, okay.
I have fuel. This luciferin molecule undergoes a chemical reaction with oxygen, essentially. It's oxidized. When this oxidation happens, the luciferin molecule jumps to a higher energy state, becoming excited. Then, as it relaxes back down to its ground state, it releases that excess energy as a photon of light. That's the glow you see.
Okay, luciferin plus oxygen equals light. But that reaction doesn't just happen spontaneously, right? It can't be that simple.
Not efficiently in a biological system, no. This is where the second key player comes in, an enzyme called luciferase.
Luciferase. Okay, so an enzyme catalyst.
Exactly. Luciferase acts as a biological catalyst, facilitating and controlling the oxidation of luciferin, making the light production rapid, efficient, and importantly, often triggered only when the organism needs it. The sources mention that researchers figured this out decades ago, demonstrating this ATP-dependent heatless light production.
ATP, that's adenosine triphosphate, the main energy currency used by cells. So the organism might expend some cellular energy to get the reaction going. Is that involved?
For many systems, yes, it's not entirely free energy. The light is released during the conversion of deluciferin to oxyluciferin, and this reaction is typically fueled by components like oxygen, certainly, but also ATP, magnesium ions, and of course the luciferase enzyme to drive it forward efficiently. It's a complex biochemical process, but the core idea is a catalyzed oxidation releasing energy as light.
That sounds fascinating. How is that different from something like fluorescence? We see fluorescent things everywhere. From high-visibility vests to molecules used in labs, they seem similar.
That's a really important distinction, and the sources highlight it clearly. Fluorescence requires an external light source.
Right, you need to shine a light on it first.
Exactly. You shine light of a specific wavelength onto a fluorescent substance. That light excites its electrons. As those electrons drop back down, they re-emit light, usually at a longer, different wavelength. Crucially, when you turn off the external light, the fluorescence stops immediately. It depends on that incoming energy.
Right. It's like the substance is reflecting and shifting the color of the light you provide. It doesn't make the light itself.
Precisely. Bioluminescence, on the other hand, generates its own light internally through that chemical reaction of luciferin and luciferase reacting with oxygen. It doesn't need any external light source whatsoever to produce its glow.
The organism is the source of the light.
Correct.
Got it. Bioluminescence is internal light generation. Fluorescence is external light-driven re-emission. And we know fireflies are the classic example of bioluminescence, but you mentioned it's found in a vast range of organisms.
Oh, it's everywhere. Seriously. The sources show this isn't just a niche trait. You find bioluminescence in worms, insects, crustaceans, fish, squid, jellyfish, brittle stars, sea cucumbers, fungi, bacteria. Even some plants in theory, although less common naturally. It's evolved independently multiple times across the tree of life, which is fascinating in itself.
Multiple independent origins, that tells you it must be pretty useful.
Absolutely. And while it's spectacularly common in the ocean, as we said, you do find it in terrestrial environments too, like the familiar firefly or glowing fungi in forests. It's not just an aquatic thing.
It's incredible how many different life forms have converged on this same solution, this ability to generate light.
Indeed. And the sources briefly touch on other ways organisms might produce light or radiation, electroluminescence, thermoluminescence, things like that, but clarify that the focus for this deep dive, especially concerning medical applications, is specifically on chemiluminescence and overwhelmingly bioluminescence, because that's where the vast majority of research and application lies, according to the material we have.
Perfect. So we've covered the chemical what. Let's move to the where. You gave that stunning statistic about the deep ocean. Tell us more about the prevalence and how it differs by habitat.
Yeah, that MBRI research using advanced ROVs and these super sensitive low light cameras is really illuminating here, pardon pun.
Nice one.
They systematically observed animals in different ocean zones. In the midwater column, that vast open water space from near the surface down towards the seafloor, they found that over 3/4, specifically more than 75%, of the animals they saw could produce their own light.
3/4. That's far more than most people would intuitively guess. It's almost expected down there, isn't it?
It really is. It's the majority rule in the midwater, and the sources state that this ratio remains remarkably consistent throughout the midwater, staying high from the surface layers accessible to light all the way down to 4,000 meters depth where there's absolutely no ambient light from above.
So it's a dominant strategy across the entire midwater environment, regardless of depth within that zone. How does that compare to life right on the bottom, the deep sea floor?
That's where the picture changes significantly. The recent research cited estimates that only about 40% of animals living directly on or just above the deep sea floor are capable of producing light.
Wow, that's a huge drop from over 75% in the open water to around 40% on the bottom. Why? Why do the researchers think there's such a difference between the midwater and the seafloor?
They propose several compelling reasons, and they're tied directly to the nature of the habitat. The midwater is essentially a three-dimensional environment. It's vast, open space, and light signals can travel relatively unimpeded over significant distances, which makes bioluminescence really effective for communication, or luring prey from afar, or maybe scaring off predators in a wide area.
Like signaling across an empty plain, basically. Your light travels.
Exactly. Now contrast that with the deep sea floor. It's a much more restricted two-dimensional space, fundamentally. It's punctuated by physical structures like rocks, canyons, sediment mounds, obstacles. And as one source notes, the water can sometimes be murkier near the bottom, either naturally from currents stirring sediment or potentially impacted by human activities like deep-sea drilling or mining causing turbidity.
Right, so if you're trying to signal with light on the seafloor, your signal is much more likely to be blocked by something or just diffused by cloudy water. It wouldn't travel as far or be as visible.
Precisely. The effectiveness of long-distance light signaling is reduced. Another hypothesis they discuss relates to the frequency of, well, non-threatening disturbances.
Like just bumping into things.
Kind of. On the seafloor, animals might be bumped or jostled by non-threatening events more often. Maybe a piece of debris drifting by, a current shifting sediment. If an animal's defensive glow is triggered by any physical contact, Doing so too frequently could be a significant waste of energy compared to a midwater animal, which might only experience physical contact during a genuine predator encounter.
Ah, that's a smart energy conservation strategy. You don't want to light yourself up and give away your position and use that energy unless it's absolutely necessary.
Exactly. It suggests a kind of cost-benefit analysis for the trait based on the specific environment. It might just be too costly to glow frequently on the seafloor.
Makes sense, but you said 40% still glow down there, so it's not absent.
Not at all. And even on the seafloor, the sources point out that within certain groups that thrive there, bioluminescence is still incredibly common. For example, within sea cucumbers, brittle stars, sea pens, those things that look like old-fashioned feathered pens sticking out of the sediment.
Yeah, I've seen pictures. They're weirdly beautiful.
They are, and bamboo corals too. Within those groups, the researchers found that nearly all tested species were capable of making light.
So if you're a sea pen or a bamboo coral on the deep sea floor, chances are you glow.
According to the sources, very high chances for the species they've actually tested. But conversely, there are entire groups on the seafloor where bioluminescence seems to be completely absent, at least based on current knowledge.
Like which ones?
No sponges, sea urchins, or sand dollars are known to glow. Though it's important to add the caveat, as the researchers do, that not every single species in these groups has been tested, so there's always a possibility of a rare exception waiting to be found. You never know.
True, the deep sea still holds secrets. But that's interesting. It's not a simple gradient of less light as you go down, but rather a complex picture influenced by the specific microhabitats and, I guess, the evolutionary history of different groups.
Exactly, and you see this even within the same large animal groups that inhabit both zones. For instance, the sources say over 90% of worm species found swimming or drifting in the midwater are bioluminescent.
90% of midwater worms.
Yeah, but very few seafloor worm species possess the trait. Similarly, about half of midwater fish and squid species glow compared to very few seafloor fish and squid. It really emphasizes how the specific pressures and opportunities of being in the open water versus being on the bottom drive the presence or absence of this trait.
That's fascinating. So it's not just about being in the dark, it's about how you live in the dark. Now for the big why. What are the actual functions of this light in the wild? What are these creatures using their internal light bulbs for?
The sources describe several proposed reasons, and they generally fall into a few key categories related to survival. Basically, avoiding, becoming food, finding food, or communicating.
Defense, dinner, or conversation? The essentials.
Pretty much sums it up. And with new sensitive cameras allowing researchers to observe these behaviors in their natural habitat via ROVs, we're getting clearer examples than ever before. They're seeing animals use their light to lure prey towards them, emit bursts of light or sparkling clouds to distract or startle predators, or use specific patterns of flashes for communication between members of their own species, perhaps for mating or schooling or identification.
Seeing those behaviors must be incredible after only guessing for so long. The sources put a particular spotlight on defensive mechanisms, especially involving sea cucumbers. Can you elaborate on those? The burglar alarm strategy sounds amazing.
It truly is one of the most vivid examples. One source describes the deep-sea sea cucumber, Enypniastes eximia, sometimes called the pink sea pig, or swimming sea cucumber, because it can actually swim upwards off the bottom, which is unusual.
A swimming sea cucumber.
Yeah, it's quite something. This species has remarkable expandable skin that contains these granule bodies that light up when stimulated mechanically.
So if something touches it, it glows, like a little warning light.
Yes, a light touch causes a localized glow just where it was touched. But if it's grabbed or bitten by a predator, the mechanical stimulation is much stronger, and the bioluminescence response is much more intense and can actually spread across the entire body.
Okay, full alarm mode.
Exactly. And this is where the burglar alarm concept comes in. This sea cucumber skin is quite fragile. Under strong attack, it can deliberately detach or slough off parts of its skin.
It just lets go of glowing pieces of itself, seriously.
Exactly. It leaves behind a cloud of intensely glowing skin particles floating in the dark water.
Wow, what does that do? Just distract the predator.
It might distract, but the main idea is more cunning. In the absolute darkness of the deep sea, this sudden, brilliant cloud of light doesn't just confuse the predator, it dramatically illuminates the predator itself.
Ah, so it lights up the attacker.
Yes. And why is that a defense? Because that predator, now brightly lit, becomes a very obvious target for its own predators, the bigger fish or squid lurking nearby.
Oh, that's brilliant. It's revealing the attacker to higher-level predators. Hey, everyone, look! There's a tasty meal right here, lit up like a Christmas tree, and it's currently trying to eat me.
Precisely. It's a sacrifice of skin to reveal and potentially expose the attacker, giving the sea cucumber a chance to escape while its attacker is potentially getting eaten or at least scared off.
That is an incredibly sophisticated strategy. Does the skin grow back?
Apparently, yes. The sources also note that laboratory studies showed this detached glowing skin can regenerate quite rapidly, so it's an expendable but renewable defense mechanism.
That's just astounding. Are there other defensive glows in sea cucumbers mentioned?
Yes, the sources mention other patterns too. Panicia moseley, another seafloor sea cucumber, doesn't slough its skin, but it produces these waves of blue and green light that travel along its body when disturbed.
Waves of light, like a ripple effect.
Yeah, like a moving pattern. And scotoplanes, the more classic sea pig, sea cucumber with the prominent tube feet that lives on the abyssal plains, also glows from head to tail in a similar wave-like pattern when disturbed.
Waves of light rippling down its body.
Yeah.
So not necessarily shedding light, but creating a moving, possibly disorienting or warning light show.
Exactly. Different species, different tactics. And it's important to note, while sea cucumbers have other defenses, like ejecting sticky tubules or even, eviscerating their internal organs.
Yeah, the really gross stuff.
Right, that. The sources we have specifically highlight the bioluminescence as a distinct active defense strategy in these particular species, separate from those other, sometimes more drastic methods.
It's just amazing the complexity of life in these environments we barely see. Okay, let's shift gears a bit and go deeper into the science behind the glow. We talked about luciferin and luciferase, the fuel and the enzyme. Is it always the same chemicals doing the job?
No, definitely not. That's another layer of complexity. While cholinterazine is a very common luciferin used by many, many marine organisms to produce blue light.
Blue makes sense, travels furthest in water.
Exactly. But nature has evolved diverse biochemical pathways. Different groups use different chemicals, leading to variations in the color and characteristics of the light produced.
Like the yellow light you mentioned earlier, from that worm.
Yes, that's a perfect example from the MBARI source, the worm Tomopteris. Most deep-sea bioluminescence is blue or blue-green for that reason, water penetration. But Tomopteris makes this distinctive yellow light. Researchers wanted to understand how. This involved the painstaking process of collecting these relatively small worms using ROVs, literally thousands of meters down, then purifying the luminous fluids they release when disturbed.
Wow, that sounds incredibly difficult.
It is. High-tech deep-sea biology. They discovered that a chemical called aloamodin, which is actually found in some plants like aloe vera, is somehow involved in generating this unique yellow light in the worm. They're still identifying all the proteins and cofactors in the pathway, but it clearly shows the diversity out there. It's not just one recipe for light.
Yeah, it's incredible dedication, collecting tiny glowing worms from the abyss to figure out their chemistry. And it paid off with a totally different system.
It did. And the sources note there's an ongoing global search for new bioluminescent systems from diverse organisms finding novel luciferins and luciferases. They mention things like polynoidin from scale worms or the unique mechanisms in fungi that produce green light. The reality is we've only fully characterized a small fraction of the bioluminescent systems known to exist. There's potentially a huge chemical library out there.
So there's a vast library of natural light-making chemicals and enzymes out there waiting to be discovered and understood. That's exciting.
Precisely. And that leads directly into the idea of engineering these systems. Researchers aren't just studying them, they're actively trying to improve them for specific uses.
How so?
For example, they've engineered railroad worm luciferase, a different kind of luciferase than found in fireflies or most marine life, to have augmented properties. They can make it more thermostable, meaning it functions better at different temperatures, like body temperature for medical studies. They can give it attenuated decay so the light signal lasts longer, making it easier to measure, or modify it for higher penetration into tissues or cell membranes. These enhanced versions are far more powerful as research tools than the basic natural ones sometimes.
Okay, so we're taking nature's blueprint and optimizing it for our specific needs in the lab. What about the genetic side? How do organisms know how to build these light-making systems in the first place?
That's where genomics and transcriptomics are really revolutionizing the field. By sequencing the genetic material, the full genome or the transcriptome, which shows which genes are actually active even from very small tissue samples of rare deep-sea animals. Researchers can identify the genes responsible for coding for the luciferases, the proteins involved in synthesizing luciferins, and any accessory proteins needed for light emission or storage or regulation.
It's like finding the assembly instructions in the organism's DNA, even if you can't easily study the organism live.
Exactly. And this genetic information allows for comparative studies. The MBARI source mentions genetic research on the bioluminescence genes of the vampire squid, which is a fascinating creature, or the photoproteins, another type of light-emitting system, slightly different chemistry, in deep-sea radiolarians, tracing the genetic origins of common molecules like coelentrazine and mapping how bioluminescence genes have evolved independently multiple times in different invertebrate lineages like jellyfish or arrowworms.
Tracing the multiple origins of this trait through the genes. That brings us to the bigger evolutionary question. Where did bioluminescence even come from in the first place? That PubMed source had a really interesting hypothesis connecting it to something seemingly unrelated.
It did. It's a fascinating idea. This paper proposes that the luminescent substrates, the luciferins themselves, like coelentrazine, may have initially evolved for a completely different purpose, as a defense mechanism against oxygen.
Oxygen defense, like protecting cells from getting damaged by oxygen. That sounds counterintuitive since the reaction needs oxygen.
It does seem paradoxical at first, but yes, the idea is protection against reactive oxygen species, or ROS. These are unstable molecules containing oxygen, like superoxide or peroxides, which are toxic byproducts of metabolism, especially in an oxygen-rich environment near the surface waters, for example. The hypothesis is that these luciferins originally acted like tiny cellular antioxidants, scavenging these harmful ROS before they could damage DNA or proteins.
So the chemicals that now make light were originally like the cell's internal cleanup crew, neutralizing toxic waste from using oxygen.
That's the idea. They were performing an essential detoxification function. The hypothesis suggests that the functional shift from this primary antioxidative role to emitting light might have happened when organisms began colonizing deeper parts of the ocean.
Because there's less oxygen down there, so less need for that specific defense.
Precisely. In deeper ocean layers, there's not only less light, but also significantly lower oxygen concentrations compared to the surface. This results in reduced oxidative stress on the organisms living there. If the selective pressure to use these chemicals solely for fighting oxygen damage decreased, it might have allowed for the evolution of pathways that could harness the inherent chemiluminescence of these molecules, the light produced as a byproduct of reacting with the remaining oxygen for a new purpose. Light emission.
Wow.
Possibly in specialized organs, while maybe retaining their antioxidant properties in other tissues where needed.
That's a remarkable concept, that the move into a lower oxygen, darker environment didn't just favor organisms that could make light, but perhaps enabled it by repurposing existing cellular machinery that was no longer under such strong selective pressure for its original job.
It potentially ties their requirement for oxygen in the bioluminescent reaction back to this ancient role. The intermediate light levels and oxygen concentrations in the mesopelagic zone, that twilight zone where bioluminescence is most prevalent, might have provided an evolutionary sweet spot for refining these proto-bioluminescent systems, balancing the original antioxidative function with the new light-emitting capability. It's a beautiful illustration of how evolution repurposes existing tools for new challenges and opportunities.
Absolutely fascinating. From fighting oxygen damage in ancient cells to lighting up the deep sea. And now that journey continues into a completely different realm, human health and medical research. How did this natural light show move from the ocean depths into the lab?
Well, as the Eastern Journal of Medicine source explains, it really became a powerful tool once scientists could isolate the key components, the luciferin and luciferase, and make the reaction reproducible and measurable outside the organism. Once you have a reliable light signal that you can link to a specific biological event, you can use it to track, measure, and visualize things that were previously invisible or very hard to quantify.
It's like unlocking a biological reporter system. Turn on the light when something happens.
Exactly. And it has become incredibly widespread. The source highlights its prevalence in many, many medical research fields today, especially areas like cancer research and basic cell culture studies. And with advancements in creating brighter bioluminescent systems, those engineered luciferases we talked about, and more sensitive imaging equipment, it's opening up new avenues for non-invasive research, particularly in live animals or complex cellular systems, allowing scientists to see what's happening inside without constantly disturbing the system or sacrificing the animal.
Non-invasive is a huge advantage in research, right? It allows for longitudinal studies on the same organism or sample over time, seeing how things change.
Absolutely critical for many types of study. Where specifically are we seeing bioluminescence make a big impact in medicine? According to these sources, what are the key applications?
The review article details numerous applications, and yeah, while we can't cover them all in depth, a few key areas really stand out. In cancer research, for instance, it's been transformative.
How so?
Researchers can genetically engineer cancer cells, say human cancer cells, for a mouse study to express luciferase. They then inject these cells into a mouse, creating what's called the xenograft model. By injecting the mouse with a corresponding luciferin, the cancer cells light up.
So they've essentially made the tumor glow. You can see it inside the mouse.
Yes, exactly. And using sensitive bioluminescence imaging systems, these cameras that can detect very low levels of light, they can non-invasively track the growth and spread of the tumor in the living animal over days or weeks. They can see if it's metastasizing to other organs because tiny new spots of light might appear. This is incredibly powerful for evaluating potential cancer therapies.
Right, you can test a drug and see the effect directly on the tumor's light signal.
Precisely. They can give a new drug to the mouse and literally see if the tumor glow shrinks, indicating the drug is working, or if it continues to grow or spread.
That allows for much more dynamic and realistic assessment of treatments than just looking at cells in a dish or waiting until the end of an experiment.
Absolutely. The source mentions its use in evaluating novel therapies for things like acute myeloid leukemia in mouse models. It's also used alongside traditional methods like immunohistochemistry staining tissue samples to give a clearer, combined picture of how a tumor is responding to treatment.
Combining different views for better understanding.
Yeah. Furthermore, it's used to evaluate the success of complex procedures like bone marrow transplantation. They can track the fate of the grafted hematopoietic stem cells, the cells that produce blood, by labeling them to glow. They can see if they successfully engraft in the bone marrow and start proliferating.
So tracking implanted cells after therapy, seeing if they're surviving and thriving, also using light, that's really versatile.
Yes, it's a versatile tracking tool at the cellular level within a living system. Another area where bioluminescence offers incredible speed and convenience is in infectious diseases and microbiology. Think about traditional methods for identifying bacteria in clinical samples, like urine cultures for a UTI.
Right, those can take a day or two, sometimes longer.
Which can be a frustrating delay when someone needs treatment quickly, especially with potential antibiotic resistance issues.
Definitely, faster diagnostics are crucial.
Exactly. The sources highlight novel technologies that use bioluminescence for rapid detection. One method described, called tube bioluminescence extinction technology, urine. Tube tear, kind of a mouthful.
Ah, yeah. Tube tear.
And even a more portable, cell phone-based UTI bioluminescence extinction technology, Cubette, use engineered bioluminescent reporters. These reporters are activated or inhibited by the presence of specific bacteria or fungi common in UTIs. The bioluminescence signal changes dramatically, maybe it appears or disappears, or changes color if the target pathogens are present, allowing for detection in minutes rather than days.
A rapid UTI test using glowing microbes or reporters. That's a direct, practical application that could significantly improve patient care, getting the right antibiotic faster.
It really could. And it's not just in clinical diagnostics. In research, bioluminescence is used extensively to track the progression of infections in animal models.
How does that work?
Researchers can create bioluminescent strains of pathogens, like E. coli or the parasite Trypanosoma cruzi that causes Chagas disease, and introduce them into animals, say mice or rats. Then, by imaging the animal's body over time, they can literally watch the infection spread, quantify the bacterial or parasite burden by the intensity of the light, and see how different treatments affect the pathogen's survival and distribution, all non-invasively in the same animal.
You can see the infection glowing and moving through the body. That's incredibly powerful for understanding disease dynamics and testing antivirals or antibiotics.
It offers a level of real-time insight that's difficult to get otherwise without sacrificing many animals at different time points for tissue analysis. Bioluminescence is also being applied heavily in drug discovery and screening.
Finding new medicines.
Yeah. Developing new drugs is a massive, complex process, often involving testing thousands or even millions of potential compounds to see if they have a desired biological effect.
You need efficient ways to see if a compound is hitting its target and doing something useful. High-throughput screening.
Precisely. Bioluminescence offers sensitive and often automatable methods for this. For instance, they can use genetically encoded nanosensors linked to bioluminescence. These sensors are designed to emit light, or change the intensity or color of their light when they interact with a specific molecule or when a cellular process occurs.
Like, say, the activation of a specific receptor on the cell surface, like the androgen receptor mentioned in the sources, which is important in prostate cancer.
Okay, so the light changes when the receptor is turned on or off.
Exactly. By adding potential drug compounds to cells containing these sensors, researchers can quickly screen which compounds trigger or inhibit the desired biological event simply by measuring the change in the bioluminescent signal using pleat readers.
It's like a molecular switch that lights up when the drug hits its target or affects the pathway. A fast readout.
Yes, a very sensitive and often quantitative switch. And this is where the engineered luciferases we discussed come in again, providing brighter, more stable light signals for more robust and efficient screening assays.
Makes sense. Better tools for faster discovery.
The sources also mention using variations of bioluminescence, like bioluminescence resonance energy transfer, or BRET. This gets a bit more complex but is really powerful.
BRET. Okay, what's that?
BRET involves two different molecules tagged close together within a cell. One is bioluminescent, the donor, and the other is fluorescent, the acceptor. The light energy produced by the bioluminescent donor molecule excites the nearby fluorescent acceptor molecule, causing it to emit light at its own specific wavelength. The efficiency of this energy transfer, and thus the amount of light emitted by the acceptor, depends critically on how close the two molecules are. We're talking nanometers apart.
So if you tag two molecules that you think interact with each other, like two proteins or a drug and its receptor, with these donor and acceptor labels.
If they come close together because they're interacting, you'll get energy transfer and see the acceptor's light signal.
Exactly. If they are far apart, no BRET signal. It's a way to study molecular interactions and proximity within living cells in real time.
The sources mention using BRET systems to study how endocrine-disrupting chemicals, environmental contaminants, affect human receptors like the androgen or estrogen receptor, and how they might impact the female reproductive system, providing a very sensitive way to measure these subtle but important effects.
That's another layer of sophistication, using bioluminescence not just to report activity, but to study physical proximity and interaction between molecules. Really clever.
It is, and the applications extend into fundamental biological processes like aging research. This was another area highlighted.
How is bioluminescence used to study aging?
The sources described using those ATP-dependent luciferase assays we mentioned earlier. Remember, ATP is the cell's energy currency, and the light output is proportional to ATP levels.
Right, so you can measure the energy level of a cell by how much it glows using that basic reaction.
Yes. This simple assay is widely used to study how aging affects cellular energy metabolism, for example, in oocytes, developing egg cells, looking at things like mitochondrial function and protein synthesis rates, which often decline with age. They can also use specific bioluminescent probes designed to monitor changes in mitochondrial membrane potential, another key indicator of cellular health and aging. Mitochondria are central to many aging theories.
Mitochondrial health is definitely crucial for aging. Makes sense to have tools to track that.
It is. Bioluminescence is also used to observe endogenous circadian oscillators, the cell's internal clocks in human cells, like adipocytes or fat cells, seeing how these rhythms might change or become dysregulated with age.
Tracking our internal clocks at the cellular level.
Yeah. And they can also use bioluminescent markers to track the accumulation of senescent cells, those zombie cells that stop dividing but remain metabolically active and secrete inflammatory factors, contributing to aging and age-related diseases. Researchers can engineer systems where these senescent cells specifically light up, allowing them to track their accumulation in different organs like the lung or thymus or pancreas as an animal ages.
Seeing the buildup of aging cells by their glow, it really allows researchers to visualize processes that were previously very difficult or impossible to track dynamically in living systems.
It offers a powerful, non-invasive window into these complex biological phenomena, and the review article honestly just lists so many other fields where bioluminescence is being applied. Immunity, tracking immune cell responses, muscular regeneration, dermatology, orthopedic research like implant integration, wound healing and infection control. It's truly become a pervasive and fundamental tool in biomedical research labs everywhere.
It's remarkable how unlocking the chemistry of a firefly or a deep-sea fish has provided such a fundamental tool across so many disparate areas of medicine and biology.
It really underscores the principle that understanding basic biological mechanisms, no matter how esoteric they seem initially, can lead to unpredictable but hugely valuable applications down the line. Once you can reproduce and measure a natural phenomenon reliably, you can start leveraging it in all sorts of ways.
So, looking ahead then, Given all these current applications and the ongoing discoveries both in nature and in the lab, what does the future hold for bioluminescence in research and potentially even clinical practice?
The sources paint a very promising picture. A key point they consistently emphasize is that, as we discussed, only a small fraction of the known bioluminescent systems have actually been fully characterized biochemically and genetically.
Right, the library is still largely unread.
Exactly. This means there are likely many more unique luciferins, luciferases, and reaction mechanisms waiting to be discovered, potentially from undiscovered species or unexplored environments like the deep sea or even soils. These could offer new colors of light, like near-infrared, which penetrates tissues better, brighter signals, faster or slower kinetics, or different requirements that might be useful for specific research applications.
More potential biological light bulbs out there with different features.
Exactly. Coupled with this potential for discovery, there's continuous development in the imaging technologies themselves, making detectors more sensitive, incorporating the ability to image different wavelengths of light simultaneously, multiplexing, and further augmenting the brightness and stability of the engineered bioluminescent reporters themselves through protein engineering and synthetic biology.
Pushing the limits on both the light source and the camera.
Right. The ongoing goal, mentioned across sources, is to develop smaller, more robust systems and probes that allow for even better in vivo imaging, seeing processes happening directly in living organisms with higher resolution and sensitivity. This includes everything from tracking the fate of a single transplanted cell to monitoring gene expression changes in specific tissues in real time, or even controlling neuronal activity in the brain using combinations of bioluminescence and optogenetics, as one source briefly mentioned.
Making the tools more precise and less intrusive for looking inside living systems.
Yes. The sources suggest that bioluminescence techniques are moving towards becoming more routine examination methods in certain areas. They cite things like bioluminescence immunohistochemistry, using antibody luciferase conjugates to light up specific proteins in tissue sections, as an example that's becoming more common. And with continued improvements in the reporters, the experimental methods, and the imaging devices, these powerful techniques may become more accessible financially and technically, potentially moving beyond specialized core labs and into more general research settings, maybe even down to the undergraduate education level for certain applications.
That would be fantastic, making cutting-edge tools available to more scientists early in their careers, sparking more discovery.
Ultimately, the consensus seems to be that continued fundamental research into bioluminescence, both discovering new natural systems and engineering existing ones coupled with advancements in imaging technology, will lead to more accessible, diversified, and powerful imaging methods for a vast array of medical research questions.
And it brings us full circle back to where we started, doesn't it? The importance of exploring and preserving the natural world, particularly these extreme environments like the deep sea. One source specifically mentioned how human activities down there.
Like drilling and mining.
Yeah, activities which can create turbidity. Clouds of sediment in the water could potentially disrupt these vital bioluminescent signals that deep-sea creatures rely on.
That's a crucial connection they make. If deep-sea animals rely on specific patterns or intensities of bioluminescence for finding mates, locating food, or signaling to avoid predators, then activities that cloud the water column could severely impact their ability to survive and reproduce by interfering with their light communication.
It's like trying to have a conversation in a really noisy room, but with light instead of sound.
Exactly. It highlights the need for careful consideration of the environmental impact of our industrial activities in these sensitive, interconnected ecosystems, which we're only just beginning to understand. We might be destroying solutions before we even find them.
It's a powerful reminder that exploring and understanding these incredible life forms isn't just an academic exercise or solely about finding tools for our own benefit. It's also about recognizing their intrinsic value, their vulnerability, and our responsibility.
And studying these extreme forms of life also fundamentally helps us understand the broader possibilities and resilience of life on Earth itself, what's possible biochemically.
Indeed. Each new discovery shines a light, literally and figuratively, on the complexity and wonder of the biosphere. So what does this all mean? We've journeyed from the crushing pressure and pitch black of the deep ocean depths, where creatures fashion their own dazzling light for survival, defense, communication,
Using incredibly diverse chemistry.
to the brightly lit, sterile labs of medical research, where that very same principle of living light is helping us fight diseases like cancer and infection, develop new therapies, and understand the fundamental processes of aging and cellular communication.
It's a powerful testament to how unlocking the secrets of nature's ingenuity, understanding its elegant solutions to the challenges of life, can provide surprising, powerful, and genuinely illuminating tools for scientific discovery and human health.
From the abyss to the lab bench, truly a light unto our understanding.
Well put.
As we wrap up this deep dive, here is a final thought for you, our listener, to consider. What other biological wonders, perhaps hidden in plain sight in our own backyards, or deep beneath the surface in environments we've barely explored, are waiting to unlock the next big breakthroughs in science and medicine?
What other living lights are out there?
Exactly. And how does our responsibility to continue exploring, understanding, and crucially, preserving these natural sources connect directly to the potential knowledge and tools they hold for solving future challenges?
The future is full of light, it seems, waiting to be found and understood.
Keep exploring these fascinating topics, and we'll be here for the next deep dive.